मुख में पर है रेत, रास बिरानी राखते खाया घर का खेत। दूजे ਨੂੰ उपदेश देने वाले तेरे मुंह च मिट्टी क्यों, तू अपने मुंह चो मिट्टी कढ़ के दूजे दे खाने च पाई है। और की ਜਿਹੜੀ ਗੱਲ ਆਪ ਕਿਸੇ ਨੂੰ ਸਵਾਦ ਨਹੀਂ ਆਇਆ ਉਹ ਸੁਣ ਕੇ ਵੀ ਕਿਸੇ ਨੂੰ ਸਵਾਦ ਨਹੀਂ। ਜਿਹੜੀ ਗੱਲ ਕਹਿ ਕੇ ਤੂੰ ਲਾਭ ਹਾਸਲ ਨਹੀਂ ਕੀਤਾ ਉਹ ਗੱਲ ਸੁਣ ਕੇ ਵੀ ਕਿਸੇ ਨੂੰ ਲਾਭ ਨਹੀਂ। ਹਿਰਦੇ ਦੇ ਕਪਟ ਰੱਖ ਕੇ ਵੀ ਗਿਆਨ ਦੀਆਂ ਗੱਲਾਂ ਕੀਤੀਆਂ ਜਾ ਸਕਦੀਆਂ। ਮੱਥਾ ਟੇਕਣਾ ਇੱਕ ਬੜੀ ਚੰਗੀ ਕਿਰਤ ਹੈ। ਮੱਥਾ ਟੇਕ ਰਹੇ ਪਰ ਸਿਰ ਝੁਕਿਆ ਹੈ, ਅੰਦਰੋਂ ਮਨ ਨਹੀਂ ਝੁਕਿਆ। ਝੁਕਣ ਦਾ ਮਤਲਬ ਇਹ ਹੈ ਮੈਂ ਸਰੈਂਡਰ ਕਰਦਾ, ਮੈਂ ਸਮਰਪਣ ਕਰਦਾ, ਐ ਗੁਰੂ ਤੇਰੀ ਹਰ ਗੱਲ ਮੈਨੂੰ ਕਬੂਲ ਪਰਵਾਹ। ਪਰ ਸਿਰ ਝੁਕਾ ਕੇ ਵੀ ਗੁਰੂ ਦੀ ਹਰ ਗੱਲ ਮੈਨੂੰ ਪ੍ਰਵਾਨ ਨਹੀਂ ਹੈ तो महाराज कहते हैं चलो 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 पिंडी क्या थी है जा रहे थे कुछ सोचते इस मत्था टेकर नल की क्या देखिएगा तो स्टेज नहीं पे सिर पे झुका देता है। गुरु की कोई गल माननी नहीं कि मत्था टेकना सिवाय पखंड तो और कुछ भी नहीं। सिर में कपट रख के कीर्तन भी किया जा सकता है। महाराज कहते हैं कोई ना दी देती बहुत बांस कर पापा ने मां बड़ी चक्की हुई है अच्छा लगे जिना अंदर मैं कुछ पापा ने बोल जांदी है जिना रोए क्या सी ਇਹਦੇ ਨਾਲ ਕੀ ਹੈ ਜਿਹਦੇ ਵਿੱਚ ਹੰਕਾਰ ਰੱਖੇ ਕਿ ਕਈ ਗੁਣਾ ਵੱਧ ਲੈਣ ਦੀ ਭਾਵਨਾ ਰੱਖ ਕੇ ਦਾਨ ਵੀ ਦਿੱਤਾ ਜਾ ਸਕਦਾ ਹੈ। ਦੂਜੇ ਦੇ ਮੂੰਹ ਦਾ ਪਿਆਰ ਸਤਿਆਂ ਮਨ ਸੰਤੋਖ ਉਪਜੇ ਦੇਣੇ ਕੇ ਵਿਚਾਰ ਦੇ ਮੰਗੇ ਮੇਰੇ ਦੇਣਾ ਨਾਲ ਲੈਣਾ ਜੁੜਿਆ ਹੋਇਆ। ਜਿਸ ਦੇਣੇ ਨਾਲ ਲੈਣਾ ਜੁੜਿਆ ਹੋਵੇ ਦਾਨ ਨਹੀਂ ਹੈ ਬਵਾਲ ਵਿੱਚ ਦੇਣਾ ਮੈਂ ਉਧਰ ਹੀ ਰਹਿਣਾ ਮਹਾਰਾਜ ਕਹਿੰਦੇ ਕੋਟਮ ਦੇਖੋ ਗੰਦਾ ਸੇਵਕ ਔਰ ਸਗਲੇ ਵਿਵਹਾਰੀ ਕਰੋੜਾਂ ਕੋਈ ਕਦਦਾਨੀ ਗੁੱਸਾ ਦੇਣਾ ਕੋਈ ਕਰਦਾ ਜਿਹਦਾ ਦੇਣਾ ਹੈ ਦੇਣਾ ਕਹਿੰਦੇ ਨੇ ਇੱਕ ਕਪੀਸ਼ਵਰ ਦੇ ਫਰਿਸ਼ਤੇ ਬੜੇ ਖੁਸ਼ ਹੋਏ, ਦੇਵਤੇ ਬੜੇ ਖੁਸ਼ ਹੋਏ, ਪ੍ਰਗਟ ਹੋਏ ਤੇ ਉਸ ਫਕੀਰ ਨੂੰ ਕਹਿਣ ਲੱਗੇ ਤੈਨੂੰ ਵਰ ਦੇਣਾ ਹੈ ਤੂੰ ਜਿਹਦੇ ਸਿਰ ਤੇ ਹੱਥ ਰੱਖੇਗਾ ਨਾ ਧਨਵਾਨ ਹੋ ਜਾਏਗਾ, ਗੁਣਵਾਨ ਹੋ ਜਾਏਗਾ, ਮੂਰਖ ਹੈ ਤੇ ਸਿਆਣਾ ਹੋ ਜਾਏਗਾ, ਅੰਧਾ ਹੈ ਤੇ ਉਹਨੂੰ ਅੱਖਾਂ ਮਿਲ ਜਾਣਗੀਆਂ, ਗੂੰਗਾ है तो उन्हें सवान मिल जाएगी, बे औलाद है उन्हें औलाद मिल जाएगी, शक्तिहीन है तो बड़ा संपन्न हो जाएगा। पति तेनू ये वर देंगे फकीर कहने लगा मैं नहीं कहना ये वर मैं नहीं कहना। क्या तू नहीं चाहता कोई मूर्ख सियाना बन जाए, कोई अंधा सुजाता हो जाए, कोई निर्धन धनवान हो जाए? क्या तू नहीं चाहना? मैं चाहता। फिर तू ये वर साढ़े को क्यों नहीं लेगा? मैं ये नहीं चाहता कि मैं जिसे सिर से हाथ रखा वो धनवान हो जाए, वो सुजाता हो जाए, वो सियाना हो जाए। नहीं, मैं जितों लंघ जावा मेरी छाया नाल लोकी सियाने हो जाए, धनवान हो जाए। मैनू नहीं पता चलना चाहिए कौन धनवान हो गया, कौन सुजाता हो गया, कौन महान हो गया। दुनिया में उन्होंने लोगों ने महान भलाई की है जिन्होंने कभी आप प्यार भी नहीं आया असं भलाई की थी। इन्होंने कहते दान नहीं। बाकी गुरु अर्जन देव जी महाराज कहते हैं कोट मदे को बिरला सेवक और सगले व्यवहार। कर्म श्रेणी के चार भाग बनते हैं तो खुद व मन सब इस श्रेणी असी किस हिस्से से खड़े। पहला कृत भी ठीक, नीयत भी ठीक। दूजा नीयत ठीक है, कभी कभी कृत गलत हो जाए। तीजा कृत ते सारी ठीक है, समाज में रहना है तो कृत से ठीक रखना, नीयत ठीक नहीं। चौथे ना नीयत ठीक ना कृत ठीक, ना सोचनी ठीक ना करनी ठीक, खुद ब मुन सब सुख खुद मुन सब बन खुद इंसाफ कर तू कौन इन्हां चारां विच किहड़ी श्रेणी च परमात्मा दे दर ते मानसिक कर्म दी विचार होंदी करमे करमे होए विचार सच्चा आप सच्चा दरबार कीता होया कर्म मन दे च लिखिया जांदा है किहड़ा मन दे च लिखिया जांदा है जो मन करके कीता जांदा पुण्य पापी आखण नाहि कर कर करना लिख ले कर्मां विचों सिरमौर कर्म है कि प्रभु का नाम जपना सिरमौर धर्म है सिरमौर कर्म है सगल धर्म में श्रेष्ठ धर्म हर को नाम जप निर्मल कर्म एक 2 मिनट में पढ़े वो लैंड ज्यादा नहीं अक्सर लोग पूछते हैं नाम जपन का कोई ढंग कोई विधि विधान पहले मैं पंक्तियां पढ़े सामने रखते हैं तज फलम कर्म विद निषेध राम नाम लेहि गुर प्रसाद जन कबीर राम कर सनेहि विधि निषेध कोई विधि नहीं कबीर कहते कोई विधि नहीं है भगतां विचों सिरमौर भगत श्री कबीर कहते कोई ढंग नहीं मैं ठंडा पानी का गिलास किसी के अगों रख दिया अगला ये आके पीण का ढंग दसो जरा गल स्पष्ट हो गई नो प्यास नहीं लगती ऐसा ढंग कुछ मैं कोल्ड ड्रिंक रख रहे हैं शरबत रख रहे हैं अगला आके तुझी शरबत का गिलास के अगों रख दिता है जरा पीण का ढंग भी दसो तो गल स्पष्ट हो गई इनो प्यास नहीं लगती 36 प्रकार का स्वादी भोजन थाल विच परोस के किसी के अगों रख दईए ते अगला कहे जरा छकण का ढंग दसो तो गल स्पष्ट हो गई नो भुख नहीं लगती भूखे खावत लाज ना भूखा से खाना शुरू कर ढंग जो पूछते हैं अजय प्यास नहीं पैदा हो अजय साचे नाम की लागे हो के भुख नहीं पैदा के तड़प नहीं पैदा तड़प ज्यों पैदा होती है कि जबान वाहिगुरु वाहिगुरु जपन रसना नाम जपो तब मतिए इन वेद अमृत भाव 2 तरह की खुराक है एक भोजन एक भजन भोजन हथ कमाते ने कामकार हथ करते हैं भजन जबान करते हथ नाल कमाया होया भोजन हथ नाल पकाया होया भोजन मुंह खाते हैं जबान नाल कीता होया भजन वाहिगुरु वाहिगुरु जपया होया इनू कन छकदे कि कन्ना दे राही अंदर जांदा जे को खावे जे को पहुंचे कोई खावे ते फिर हजम करे और मैं अर्ज करा बाहर की दुनिया च रोटी कमानी औखी है रोटी पकानी भी औखी सेवा तो उन्हें बिठती जा रही है अब हलवाई लंगर बनाते हैं कई तो जाते हैं गुरु का लंगर मैं तो 99% देखा है हलवाइयों का ही लंगर सेवा हलवाइयों की गुरु का लंगर किस तरह होया तो सेवा नाल बनता है गुरु का लंगर होता तो है नहीं जिथे सेवा की भावना नाल बने वो गुरु का लंगर उसका स्वाद भी कुछ और होगा वो प्रसाद हो गया वो जिंदगी में बदलेगा वो गिणतियां कोई और तासीर हो गई है मैं इस गल में लमका मन और तसली है अर्ज करना और ये हकीकत है सच्चाई है बाहर के जगत में रोटी कमानी पकानी औखी है खानी बड़ी सौखी कोई नहीं आता ऐसा ऐसा तो सारे कहते हैं कमाने थक गए पकाने थक गए सुने किसी को खांदे थक गए लेकिन मैं अर्ज करा भजन करना सौखा वाहिगुरु 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 बन गया वाहिगुरु नाम का भोजन बन गया तेन छकना ही देखो खावे कोई खावे खांदा नहीं क्योंकि वास्ते बड़ी एकाग्रता होगी तो मनुष्य सुन सकेगा वरना नहीं। गाविए सुनिए मन रखिए सरवणन गोविंद गुण सुनो हर गाओ रसना की गवाओ वाहिगुरु का अलाप करे ते मन करना देना ही सुने ज्ञान वो फिर वाहिगुरु की धुन मन में प्रवेश करेगी। जैसे जैसे मन में प्रवेश कर दी जाएगी मन जागता जाएगा। जब जब मन पूरा जाग गया परमात्मा प्रकट हो गया, मन परमात्मा का रूप हो गया। इन बाकी क्लासा में सवेरे की कथा करांगा। सिर्फ हूँ पंज लगातार वाहिगुरु का उच्चारण करता है। मैं हमेशा मैं आलू करना इन मेरी पोकी निर्माण का भी न करना बिल्कुल हो न समझे हर जगह में कहना पता कर मैंने इतना समा हो कथा से मैं रोज कई दफा होतियां की प्रेरणा करके दिन दिन 3 जगह 4 जगह सवेरे में
So, hello students, good evening. So today, so how are you all of you? I hope you are all doing good, doing well. Good evening everyone. And I'm just concerned that is my pictures coming fine? Is there any buffering happening? If sound is coming okay, because today I'm connecting it with my mobile so that wherever I go I can connect to the internet via my mobile there also. So I found this opportunity. Today I took lectures already with genetics via my mobile. So even though I have Wi-Fi at my home, but I'm still able to uh, take my okay, good, all is okay, so everything is fine. So I will not waste much time. So it might happen that all the data might lose. So I have limited data to stream. So I will try my best to send you everything. So I will send you first everything so that at least you have data with you. So it will be recording again today from now onwards. So there was some glitch from YouTube itself, but it started working now. So from yesterday, I want to send you how to isolate protein, some protocols of that. So I forgot to send you this. And today's lectures also. So I'm sending you the protocols of gel electrophoresis and today's lecture, these two parts. So everything has been shared with you. Go through them, and okay, then I will not wait much. So I will start now. Um, so today we're gonna discuss about electrophoresis. So in which we will discuss about various types of electrophoresis and how these techniques are done. So theory of electrophoresis, mainly method to analyze various compounds and capable of acquiring the electric charge in a conduction electrodes. So electrophoresis may be defined as a migration of charged particles through a solution under the influence of an electric field. So ions that are suspended between two electrodes tends to travel towards the electrodes that bears opposite charges. So there are two types of electrophoresis, that is zone electrophoresis and moving boundary electrophoresis. So zone electrophoresis includes four subtypes, that is paper electrophoresis, gel electrophoresis, thin layer electrophoresis, cellulose acetate electrophoresis. Whereas moving bound electrophoresis takes place in four types, that is capillary electrophoresis, tacphoresis, isoelectric focusing, and immunoelectrophoresis. So out of them, mainly in the lab, it depends upon what you're looking for, what is your basic needs are. So according to that, you have to use things. So you can use capillary electrophoresis, isotachphoresis, isoelectric focusing. It's up to you, whichever you want to use it. So zone electrophoresis includes it involves a migration of charged particles on the charged particles on sporting media, and it has paper cellulose, acetate membrane, starch gel, polyacrylamide. So all these comes into the zone electrophoresis. So the main advantage is that they are useful in biochemical investigations. Small quantity of samples can be analyzed. Cost is low and easily maintenance. Whereas the disadvantages are that they are unsuitable for accurate mobility and isoelectric bond determination. And due to the presence of sport medium, technical complications such as capillary flow, electro-osmosis, absorption, moving molecular sievings are introduced into them. So general methods of operation includes saturations of media with the buffer, sample application, electrophoretic separation, removal of the sporting media. So all these four parts come under the general method of operation. So under the instrumentation, electrophoretic chamber are included, electrodes are needed, diffusion barriers are needed, sporting and stabilizing media. So these are the inert to sample and to any development reagents. So paper electrophoresis, It includes, we have electrolyte, and they are attached to the 45 volt in DC, 5 batteries of 9V and 2 glass plates. And it is a pencil line there in center with the paper filters. And these are the apparatus for paper electrophoresis, and these are the horizontal paper electrophoresis, and they are vertical paper electrophoresis. There are two types. So, and these two types are air tightening. So they are airtight first of all, and they are positive and negative side in both cases in the horizontal and vertical paper electrophoresis. And in the horizontal, you have horizontal space like this on the top, and then on the vertical it's somehow on top with this triangle shape, and we have filter paper strip on this top and sample application point to be done here. So we use Wattman number 1, number 3 millimeter in strip of 3 or 5 centimeter. Wide has been used to good effects. So separations takes place 12 to 14 hours, and it is economical and easy to use. So disadvantages are certain compounds such as protein hydrophilic molecules cannot be resolved due to the absorptive and ionogenic properties of paper, which results in tailing and distortion of the component bands. So this is continuous electrophoresis, where you have buffer solution, a sample reservoir, electrodes, and then filter paper, sample components, and they are being collected like this. So in three ways. So each there is sample collection at the bottom, and you can collect them. So used for preparative scale purposes, predetermined sample volume of 0.2 ml per minute through the wall device is applied continuously in the center of the paper. Silica gel, powered glass sand, and agar gel can be used in this case. So potential difference of 500 volt is applied with this positive and negative electrodes, and pure compounds are collected in separate containers, and the solvent is evaporated and pure fractions are reused. Then comes the gel electrophoresis. Is brought through the molecular sieve technique based on the molecular size of substance, and gel material act as a molecular sieve in this case. So gel is colloid in solid form with 99% is water. It is important that sport media is electrically neutral in that case, and different types of gel can be used in the like agar and AGAROSE gel, star, Cefadex, polychromate gels. So we have done this gel electrophoresis as a workshop or not? So we have not done that thing. So I will show you gel electrophoresis as a workshop also, where everything is done from how to make gel and how you sieve your samples, and then you do attach to your power supply. So there are two types again, vertical and horizontal starch gel electrophoresis. And agar and AGAROSE gel. Agar is a mixture of polysaccharide extracted from the seaweeds, and AGAROSE is a highly purified uncharged polysaccharide derived from agar. And AGAROSE is a chemical basic disaccharide repeating units of 3, 6 anhydro and galactose. So AGAROSE glucose dissolves when adding to the boiling liquid. It remains in a liquid state until the temperature is lowered to about 40 degrees Celsius, at which point it's become gels. And then AGAROSE gels are fragiles. They are actually hydrocolloids, and they are held together by the formation of weak hydrogen and hydrophobic bonds. Their advantage is that they are easy to prepare. Small concentration of agar is required. Resolution is superior to that of filter paper. Large quantities of proteins can be separated and recovered, and adsorption of negatively charged protein molecule is negligible in this case, and it absorbs protein relatively less when compared to other medium. Sharp zones are obtained due to less adsorption, and recovery of protein is good, and good methods of preparative purposes are done. So electroosmosis is high, that's a disadvantage. Resolution is less, and different sources of a batch of agar tend to give different results and purifications often necessary. And application is that they are widely used in immunoelectrophoresis. Gel structure of agarose looks like this. There is a solid state, then initial gel and final gel structure. So initially you make a temperature of 45 degrees Celsius, then that goes to with the aging, it make a final gel structure. First is the initial gel, then it's make a final gel structure. It could go come back with 100 degrees Celsius. If you make around 100 degrees Celsius boiling temperature, they could come to the solid state again. So something about gel electrophoresis.
This video will take you through the process for loading and running DNA samples on an agarose gel. You will be using a mini subcell to perform agarose gel electrophoresis. The subcell has an electrode wire running across the bottom of each end. This provides for an electric current to pass, which separates the DNA fragments in the samples. First, align the gel so that the wells are closest to the negative or black electrode. DNA is negatively charged and will move from the negative electrode through the gel toward the positive or red electrode. Place the agarose gel into the gel chamber. Add electrophoresis running buffer to the reservoirs at each end of the gel chamber, and keep adding buffer until the wells in the gel are covered by at least 2 millimeters of buffer. Place your samples to be loaded in the correct order according to the lanes they are assigned to be run in. Check your lab protocol for this information. To obtain a DNA sample, slowly depress the plunger first or soft stop. Holding the plunger down, insert the tip of the pipette into the microtube with the DNA sample. Place the tip close to the bottom of the sample, then slowly release the plunger button. When loading the samples, keep the pipette tip perpendicular to the row of wells. This will reduce the risk of accidentally puncturing the wells with the tip. Lower the tip of the pipette until it breaks the surface of the buffer and is located just above or just inside the well. Slowly apply pressure to the plunger button and observe as the sample fills the well. Make certain you stop at the first stop on the pipette, pause, then slowly remove the pipette while keeping your thumb down on the plunger. Once all the samples have been loaded, avoid any bumping or movement of the gel chamber. This might result in the sample spilling into adjacent wells. Place the lid on the gel chamber with the terminals correctly positioned to the matching electrodes on the gel chamber, black to black and red to red. Connect the electrodes to the power supply, again making certain the electrodes match the terminals on the power supply, red to red and black to black. Switch the power supply on, then set the correct constant voltage for running the samples. See your lab protocol for this information. If a timer is available on your power supply, set the clock for the proper time for your run. Press the start button to begin the flow of current that will separate the DNA fragments. At this point, you should be able to see bubbles coming from the wires at each end of the gel box. Notice that the positive or red electrode has fewer bubbles than the negative or black electrode. In a few minutes, you should see the samples begin to migrate from the wells into the gel. Notice as your DNA samples run, the dye front moves from the negative electrode towards the positive electrode.
So that was about Biorad AGAROSE gel electrophoresis. I will repeat, bache. So first, um, yeah. Mainly in your lab, you have to go through gel electrophoresis that I have shown you the video. That most likely you will be doing. Others are not being used. So this one, electrophoresis. Let's start. So our DNA has shorter, middle and longer DNA fragments, and they look like shoelaces, and we need to sort them out now. How we can sort them? So this is our DNA strands are molecules that are tiny, which cannot seen by microscope. So we need to check them with the help of some other technique. So that technique is called as gel electrophoresis. So this has our DNA, and scientists has used gel electrophoresis whenever they need to sort DNA strands according to the length, and this technique used for separating other types of molecules like protein also. So how does this technique works? So basically you have these SDS gels or polyacrylamide gel, and these gels have jello O shape like many holes in them, and you pour your samples on top, and we place DNA sample into holes at one end of the gel. Then you have electrophoresis. This is how you push your DNA through the gel filter by adding on electric current, and we can make it make the DNA move. So by moving, so you can see each sample has different fragments been cutted out. Some are small, large, large, small, middle, small, large, three or both, middle and large. This is how they are all separated. Then staining the sorted den. We cannot see them like that, so we have to put them under UV light so they could come and visible like this. So let's start the real experiments. There will be 5 steps. We will make the gel, set up the gel apparatus, load the DNA sample into the gel, hook up the electric current and run the gel, and stain the gel under and analyze the results. So you need agarose, flask, gel mold, gel comb, buffer and microwave. So these are the things that we required. So first we make gel with the help of agarose. So small amount of agarose we take and add into the conical flask, conical bottom flask. Then we add a liquid buffer to it. So because of that, charges could flow. So we will put some plastic cover on tip and open the microwave and add this liquid so that it could be in the liquid state. Put it outside and then put into the chamber, this melted form. And then the reason to melt it was to add the comb on top so that we can have well, because we cannot have wells in the solid state. So that's why we made it into liquid and we add comb on the top. So we will keep it until that it's get warm, and we carefully remove them now after it's become solidify. Now set up gel apparatus. So it was making of gel is done. So now it's time to set up of the gel. So you put your buffer into the electrophoresis box, and then you add your chamber. So you have loading buffer, DNA sample, DNA standard, size standard, micropipettes, gel box with the buffer gel, and pipettes. So loading buffer is done to add into DNA sample so that our DNA could be heavier and could be visible while running on the electrophoresis gel. And DNA standard sensitides are the molecular ways through which we can confirm which size our DNA sample is. So we take the micropipette, add loading buffer into the DNA sample. So we will use this and add into the electrophoresis gel like they did in the video. Carefully. So in real life it's quite difficult sometimes initially, but if you try sometimes then it's quite easy to do so. Now we will add DNA size standard. We will plug them now, red with the red, black with the black. And turn on the power supply. So bubbles will start coming inside the chamber. That means the buffer is working out. Now we are done with the making gel, set up the gel electrophoresis and loading the DNA sample into the gel. Now we have run the electric current and run the gel. So they are migrating now. So now we will stain the gel and analyze the results. So we will put them into the ethylene bromide solution as a staining solution because it will attach to the DNA base pairs. And it takes about half an hour to stain the DNA in the gel, and we will speed up the clock. So half an hour it will take, and then we put them on the ultra UV light to check them. So we turn on it. So we can now determine now we have to determine the sizes of our gels. So this could be size of around how much, students? 1000, 2000. This could be size of, any guesses? This could be size of how much? And this could be size of how much? I think this could be of 35. This could be of 1500. This could be of 6000 base pairs. So all estimations are correct. So we have discovered, we have separated our DNA with top being at the 6000 base pair, second is by 3500, and the third is approximately 1500. So congratulations, we have run our first DNA gel electrophoresis experiments. So that's it. Just share with you link of this. So So bache, basically in this you have your samples and it seems like they have sieve tubes, and from these sieve tubes you have collection samples here. So once you run them under the electrode buffer, so under the potential difference of 500 volt, pure compounds are collected in separate containers and solvent is separated and pure fractions are reused in that case. And then filter is moved and apparatus and strain to locate the separated components afterwards. So now comes the polyacrylate gel electrophoresis that we also did yesterday from this 2D gel electrophoresis that professor was telling us about great tips from the biorod itself. So it will be continuing with that. So it is prepared by polymerizing acrylide amide monomers in the presence of methylene based acrylamide to cross-link the monomers. So they are structures of acrylamide. Polyacrylate gel structures held together by covalent cross-links, and their gels are tougher than agarose gels. They are thermostable, transparent and strong, relatively chemically inert, and gels are uncharged and prepared in variety of pore sizes. So proteins are separated on the basis of charge to mass ratio, molecular size and phenomena called molecular sieving. Here you can see how the gels looks like, and this is the structure of acrylamide, which has N structure polyacrylamide of this polypeptides. So gels are stable over a wide range of pH. In this case, gels of different pore size can be formed, and simple and separation speed is good comparatively. So there are two types of page, that is native page and denatured page. So native page is which is done on the non-denaturing conditions, that is analyte is maintained in the original state. So separation is based on the charge, size and shape of macromolecules and useful for separations or purifications of proteins. And this was the original mode of electrophoresis. So their denatured page or SDS page upon the molecular weight of proteins, the common method for determining molecular weight of proteins and very useful for checking the purity of proteins. So the SDS normal page. So it's the same method like that one. We have a buffer, then you run them over the gel. There are cathode and anodes, macromolecules are moved towards the anode, and there is no external solvent space in this. Rate of migration depends on the charge to mass ratio. So you have a buffer, gel, high voltage. So SLAB, this is SLAB. So this is polyacrylate gel is vertically between two glass plates, and proteins bands are separated on the basis of relatively molecular weight and visualized with the stains further. It's the same what we did in the SDS page yesterday. We check them by ethidium bromide and check them on the autoradiogram also. And this is SDS page, that is sodium dodecyl sulfate polyacrylate gel electrophoresis, which is quietly used in the biochemistry, forensic genetics and molecular biology to separate proteins according to their electrophoretic mobility. So when a detergent SDS is added to the page, the combined procedure is termed as SDS page, and SDS code proteins molecules giving all proteins and constant charge mass ratio. Due to the masking of charges of proteins by the large negative charge on SDS binding with them, the proteins migrate along the gels in order to increasing sizes or molecular weights. Molecules in solutions with SDS have a net negative charge with a wide pH range, and they have a chance to bound to the SDS in proportion to the relatively molecular mass. So the native protein is there, we heat redundant and add with the SDS, and they get attached to the denatured protein with the bounded SDS, so like this. So there is N side, carbon side, and then SDS is attached on top of it. So native protein is unfolded by heating in the presence of beta mercapthanol with SDS, and SDS binds to the protein so that it stays on the solutions and denatures afterwards. So this we did on the fourth day of our lecture, that how different proteins are being separated according to the different sizes, and we have discussed this already also before based on SDS. So, and further, starch could be used instead of agar in boiled buffer to give colloidal suspension, and suspension on cooling sets as a semi-solid gel